It's, oh, on the text. Yeah. Oh, shit. I think I just started it.
No.
Hello, everyone.
And Betty.
Hey. Yeah.
We're going to get started in just a minute here. If we're ready, I hope everyone read the awesome research paper. We're just about ready to get started today, May 22nd, on our Journal Club Session 5 on the topic of culture of viable human blastocysts in defined sequential serum-free media. Before we get started, I just want to call out a few housekeeping items. Please, as if you're on the other side of this remote virtual presentation, please feel free to turn on your camera and your microphone and participate. Please feel free to submit your questions anytime by coming off camera, raising your hand, or just putting a question in the chat. And just a reminder, you will get a recording to this, the recording of this event before we are done. And now I'm going to turn it over to Dr. Tony to talk about the agenda and today's presentation.
Great. Oh, thank you, Liz. Yeah. This week, we're getting in at the IVF Academy, we're getting into what I call embryology. So we do 2 weeks of andrology. We finished up andrology last week. Our first week of embryology, where we learned to handle the oocytes and move things around. So I thought this was a great paper to bring in, just talking about media, because while I was working on my master's, this paper had come out just before I started working on my master's. And this, if you go back, is written in 1998. And many of the papers actually referenced in here were, it goes back as far as even to the 70s and 50s. So this is just a really good paper. It talks about, there's a lot of names in here. David Gardner was a pioneer in this field and he was part of developing a really great product out there that's sold by Life. And but what I want to just kind of, kind of bring everybody's attention to, he gives thanks to his mentors, Henry Lease, John Biggers, Bob Edwards, Barry Bavister, and Don Rieger. All of those guys are just legends in the field that did a lot for the early development of medias. And so this really talks about a lot about sequential medias, and a lot of labs are even doing single step medias. And so how are people, this is where it really started, from going to blastocysts, and talking about how implantation rates are only like 10%. And then he was quoting implantation rates of 50%. So a lot of people in the audience are probably like, yeah, so what? But when you have a 10% implantation rate, when someone says they got a 50%, they're going to call you a liar. And I even had people tell me when I had 50% implantation rates, because I was an early adopter to blastocyst transfer, people called me a liar. And so, but today that's not uncommon. So it's kind of nice to look back at where we were and where we're at today. So with that, I'm just going to kind of open it up to anybody. What we have here in the audience here locally is our students. I know we might even have some students that are actually online here joining us today, past students. I see some names that are familiar and some new names. So I'm really glad everyone's here. And so just open it up to some discussion. I don't want to be the only one talking about it. So, but yeah, and actually Lygia has a poll, asking me some questions. So let's jump into a poll real quick and kind of see where people are at, what they thought of the paper.
So we might keep talking as you, do it.
We just tab through or do, so tab through individually on our own.
Doug, are you still seeing the poll questions?
No, I can't see the poll questions.
Mm.
Is it on like a separate screen? Oops, sorry.
Well, it should be popping up on your.
Is it popping up on your screens, the polls?
Oh, I see it now. But it's just a check and an X. I don't have a question. Maybe it's in the chat. Oh, I see. But it doesn't tell me what I'm voting for.
That's not good. So. So we try verbally. So. This is a question for the group. Can just like turn on your camera or raise your hand. If your lab buys pre-supplemented with protein, or do you add the protein in your lab? I took a guess, so. Supplement.
Yeah, yeah, that's a good guess. Yeah, I think the first two polls is just getting a feel for what people are doing for, if you're pre-supplemented or you're adding your own protein. So let's just go ahead and, start talking about it. So as for the students here, like, that read the paper, Well, what was your takeaway message from it? What are some of the takeaways from it?
Many implications with different ways we can use media to help increase implantation.
Blastocysts.
Huh?
Types of blastocysts increase the implantation rate.
You gotta think back then in 1998, very few people were doing blastocysts. Today, almost a lot of people are doing blasts. That was one of the poll questions on here, but so to do blastocysts, you needed to use the sequential culture media. He basically says, like, you can make blastocysts, but they're not very good blastocysts. And the medias back then, you basically had salt, calcium chloride, potassium chloride, sodium chloride. You had all the chlorides, right? You had lactic acid and pyruvic acid. He talks about pyruvic acid in here a lot. He didn't really talk about lactic acid. And you had sodium bicarb to do your pH. That was really what media was. And if you took the medias that all these mentors had and you lined them up next to each other, they're all pretty much the same. And even today, a lot of them are pretty much the same. But what this paper, why I really love it, is it talks about a lot of the different things that it's needed in the media at different stages. So without me telling you, like, what were the stages that were needed? What was needed at the early stages? What's needed in the blastocyst stages, and what isn't needed in the early stages, and what isn't needed in the blast stages? So this is why they call it sequential. It's basically from day 1 to day 3, and day 3 to day 5. So what are the take-home messages from that?
I like how it it said that in order to have like a good ICM, like certain things like amino acids and HSA is needed to form a good ICM. And with medias that don't have it, you may get a blast that has absent ICM, or it's not a very great graded ICM. And then that is linked to like fetal development as well.
Right, that's I like to call it the fruit. You know, your profectoderm is your root, and your inner cell mass is your fruit. So without a fruit, you know, you're just going to have a plant growing, so to speak, but there's no fruit. Anybody in the audience, do they want to kind of, if they've read the paper, have any kind of what did they get from the early development versus later development stages? The system, he breaks it down into different pieces, like say carbohydrates. What is needed by the carbohydrates? The sugars. What's important in the early stages versus the latter stages? The glycolytic activity, he calls it.
In the early stages, it's like low glucose concentrations, like day 1 through 3, whereas day 3 through 6 or even 7, it's an increased glucose concentration.
Exactly. Yeah, there's a less glycolytic activity in the beginning than there is at the end. And so, like, if you think about it, none of us really knew any of this back then. So he did a lot of work. Like, this is what's happening in the oviduct. So he created a media around what is seen in the oviduct, right? And then looked at blastocyst development in the mouse model. Anybody think that? Anybody ever see any flaws in that? Say the mouse model.
Different species.
Different species.
I do.
Like
I mean,
exactly what humans need, like same amino acids, same nutrients, same.
Sure. Go ahead, Brittany.
you can grow mouse in Budweiser, man. They're pretty hardy, and you certainly can't do that with human embryos. I just think, in the training lab we were able to culture, mouse embryos in And we tried to not get them to grow, and they grew. So I just think, the things we could do with mouse embryos versus human embryos is just vastly different. I don't think the mouse model is a good representation of what, human embryos can do. I think it's great for embryo assessments, but as far as toxicity and things like that, I mean, I don't think it's a very good assay, to be honest.
No, I mean, but you think back, a lot of those, Biggers, Rieger, Edwards, everybody, they built it around the animal model though. And that's been a big discussion, like always, that, within the development of these, they were built around on the mouse model, and it's not the perfect model. Like the genome is activated at the first cleavage stage in the mouse versus in the human, it's not till day 3. Between that transition between day 3 and day 4 is where somewhere in there is where the genome is activated. So you can see where in that point, it was when the glycolytic activity is also changing. I thought that was interesting, and he actually looked at, intercell masses and things like that being formed. And I remember when the medias first came out, the EDTA was one of those pieces that was in the early stages of the media that you would see in the day 1 to day 3. And he talks a little bit about it in here, that as a scavenger of the kind of the free radicals in the media. So I don't know that that's in the medias today. I don't know that it is, because most of the single step medias. And you think about it, this was sequential. The media was built around what was needed from day 1 to day 3, and then day 3 to day 5. I don't want to jump too far ahead into the single step medias today, but like EDTA was in there. And, we used to make some beautiful embryos, and I think EDTA had a real big part of that. What about amino acids? He talks about some amino acids or some carbohydrates, and then he jumps into the amino acids. Anybody have what was the take home message with amino acids from the early stage to the later stage? Anybody from the audience, welcome to chime in like Brittany did. Just take your mute off and speak out.
I thought it was interesting that it said that that they have no beneficial effect at cleavage stage of embryos, which I don't know. In my head, I feel like amino acids are like within just the human body itself are like important with like cell regulation and that kind of thing. So the idea that they're not actually helping with like that cell cleavage, I don't know. I just found that interesting.
He did say that there were some amino acids that he found were more important in the early stages. That is also reflecting what's in the oviduct, in the natural reproductive system. What amino acids were those?
Non-essential.
Thank you. Non-essential amino acids. And so the non-essential amino acids were there, but the essential amino acids really were not really deemed to be that beneficial. And so in the later stage media, when you think about what's happening when your genome is activated, what's the difference between, you think about in the beginning, everything that embryo needs is basically built into what's the cell energy? What makes the energy in the cell? Which makes ATP, right? And what is that used for? In the production of, what's the building block of life? Proteins. Your DNA is making you're making protein. So everything the egg needs in the beginning is needed is in the egg. But at the genome activation, it's no longer the egg. It needs the nutrients from the soil. Like we need to bring in nutrients or we die. If you don't feed those embryos nutrients, they will die. And that's, and so that's what it needs. So in your later stages, it makes a good sense. It needs essential and non-essential amino acids. And so it's really super important to kind of keep that in mind. Because the idea is when embryos are growing and they're not growing well, every day you're working in the lab and they're not growing well, what is missing in the media? And And I would just like to just kind of like, I would be interested to know, and maybe, if people could just maybe is there a way to raise your hand? As a raise your hand, who is using sequential medias in the audience?
Watch this poll, just go up or down.
Yeah, raise your hand if you're using sequential medias. Okay, so 100% are using, well, we got four votes there. Come on, fellas, ladies. I guess I can vote. Maybe some people aren't in front of the computer too. I know some people in here aren't. Oh, wait a minute. I'm not using sequential. Sorry, I lied. So yeah, one, and that was, I lied. So I was actually 100%. So yeah, so most people are using single step medias. So just as another, can maybe do another one here, yes or no? Thumb up or thumb down. Who is buying it pre-supplemented with protein? Meaning like it already has the protein in it and you don't add any protein.
Me, I buy my media with protein HSA.
Yeah.
I've also worked at a lab where we supplemented with protein. And
Yeah.
that was typically because the lab protocols called for, well, then called for a higher concentration of protein for recovery. So we would do like a 20% media for biopsy and for frozen embryo transfers, egg thaws, because the idea was that the protein would help with the recovery. But I've been working at a lab for the last 2 years and haven't done that, and everything seems to be working perfectly. So I don't really think that it's necessary at this point, the way that I used to think. Um, I've seen great results with both methods. I just see if you have a higher concentration of protein, or if you don't, if you have a media with no protein in there, I think you can get into a lot of not so good variations. If you're having your junior embryologist, beginning embryologist, even your senior embryologist, it's very easy to grab the wrong media bottle, and if you use the wrong media on a case, you're in some trouble. So yeah, I personally think that getting the media with their protein in is the way to go. It's just my
Yeah,
opinion, though.
that's good. Thank you, Brittany. Actually, you bring up a really good point. You mentioned using the wrong media, the wrong broth, wrong protein. And whenever I used to use sequential medias when they first came out, that was one of my biggest worries, because you would buy the bottle, it might have a purple top for blast media and a pink top for the day 1 to day 3 media, the cleavage stage media. And they're all kind of slightly pink because there's some phenol red in it, or some media don't use phenol red, it's clear. And so what I was always worried about was when somebody was making media or making the dishes, would they switch the medias out? Because once you take that purple top off, it's really hard to tell. It used to be harder to tell. Today, they're actually putting that purple and that pink on the label because some people probably said something to the vendors. And so it would be real easy to mix that up. So if all your embryos are arresting, it would make sense that, hey, protein synthesis isn't occurring. People probably use the cleavage media for their blast media drops. And so that's one of the nice things I like about the one-step medias today. And so, but before we get into that, too far into that, so amino acids, non-essential amino acids are really important in the beginning, and then the full complement of essential and non-essential amino acids important towards the end. He doesn't really talk about anything in here in the context of fertilization medias. And so The fertilization medias also have glucose in it because the sperm needs that glucose to be motile. And so, you know, and most people, if anybody does any conventional, most of the people I see coming through any training, they're doing mostly ICSI. And so if you're doing ICSI, you can go straight into that cleavage stage, or if you're using your one-step media, you could go directly into that media because the full complement of everything is going to be there. But if you're doing conventional, you should probably use a media that has some glucose in it. I've done conventional, and I personally have had an experience doing InvoCells when we did conventionals, and I would use the one-step media because I was doing intravaginal culture and culturing all the way out to day 5 and on the blastocysts, and we would get great fertilization with it. So you can get fertilization with it. So amino acids, I really loved how he talked about intercell mass formation with different amino acids being present and not present. And, you know, basically talking about that. So development of the sequential medias. And I think he even mentions here in the beginning, just kind of in the introduction, that that, you know, day 2 or day 3, like, and he actually uses biological proteins. And so he doesn't really use the word maternal serum, but back in the day, we couldn't call up, you know, Cooper or Sage or Vitrolife and those guys and say, hey, send me some HSA. We had to find our own proteins. So a lot of times we would actually we would actually retrieve serum from the patient, and we would have to treat that serum. We'd have to basically let it clot, spin out the red blood cells, and pull off the serum. Heat inactivate that serum because the serum would affect the activation of the oocytes. And then what we would do is have to then filter that. So we made our own protein for our patients. You know, before some of the other proteins were available, there was a form of HSA. It was called plasminate, which actually we would order from burn units. It was the same proteins they would use for burn victims. And so we would, you know, plasma protein, we would actually use that, and found that actually had we learned some things with that alpha and the beta globulins in those proteins. And so that's when HSAs kind of came around, not too long after these sequential medias, are pretty much around the same time. So in the early 90s, a lot of our proteins were actually from the patients themselves. And I knew the CCRM people up in Colorado in the beginning. They would actually get the fetal cord serum. They would from the hospital, and they would actually process that cord serum and filter it and sterilize it. And so they had basically. So when we bought proteins like that, if we found a good batch of protein, we would buy, you know, a whole year's worth of protein, so we wouldn't have variability in our protein supplementation. And you have to have protein. There was even points in time in the history of IVF, we would use, I think he even mentions in here, BSA, bovine serum albumin, and I think he used 2 milligrams per mil in some of his studies. But bovine serum albumin is just a powdered form, and we would add that to the media. And then, you know, what the protein does is really just there as a surfactant. He doesn't really get into what is the protein doing with the medias, but it is needed. And so I wish Ellie was here because she mentioned last week, or maybe maybe in one of the recent Journal Club sessions, that some of the imprinting work, especially we knew in the animal world that when we used fetal calf serum, the proteins would actually cause large calf syndrome. And what it was an imprinting error in the embryos. And so what we would get is calves that would normally be born at 50 pounds were being born at 150 pounds, are cloned cows. And so all those cows had to be C-sectioned, which made cloning not successful because a lot of the calves didn't make it, or it was just very expensive to have these calves born. So we didn't know that. We didn't even know what the word imprinting was back then. It hadn't been discovered. was just being discovered because of those kinds of things. So we have to be careful with our proteins. Are we causing any of the errors by using too much protein? So that was her idea, is like they're saying like maybe you should be even using less protein. A lot of medias that we buy come at 5 to 10% or the concentration of 5 to 10 milligrams per mil. But do we have any that we still need protein, but should we go 20%? Anybody have any thoughts on that? Anybody have any history or experiences with that? Anybody use more than anybody use 20%? Maybe do a poll like 10%, 5%, or less than 5%. That able to do?
It's getting a blank screen full of.
It's okay. It was interesting that he talks about some protein, and I've never actually, I honestly don't remember this from the first time I read this paper years ago. But he talks about hyaluronate, and I remember seeing the word hyaluronate for the very first time when I read. It wasn't this particular paper, and he uses the term glycosaminoglycan, and I just love saying that word because it makes me sound smart when I say it. Don't you all agree? Thank you. Yeah. Thanks. Thank you. So yeah, it's a hyaluronate, but anybody ever hear of hyaluronate anywhere else?
And
Skincare.
yeah,
Skincare, yeah, and cosmetics.
it's a chemical that is all HA fillers.
Yeah, but he mentions that it's naturally occurring in the reproductive system. And so what he found was that, well, does anybody know what it says in there? Did anybody read the paper? I'm just curious because you guys are just way too quiet for this. Anybody read the paper?
Yeah.
You did? What did it say about, hyaluronate?
Next time I'll be honest, I read it this morning.
What's that?
I'll be honest, I read it this morning.
Okay, that's good. Appreciate your honesty.
Did it say something that could potentially like replace the culture media?
Okay, you definitely could replace the protein, and I know that Lygia had this as one of the polls on there. Has anybody tried using it? And I pretty much know the answer. The answer would be no. But there is what came out of the original paper. I remember reading David Gardner's paper on embryo glue. That's the best name for embryo for any media to ever be sold. Embryo glue. Patients will call up and demand the use of embryo glue because I want to glue my embryos in. I love that name. But I remember reading it and it wasn't available. I just read his paper. And so what do? I go to the Sigma catalog, and this is pre 1998. They didn't have Google back then. We had to look up, and I find glycosaminoglycan, and it was harvested from rooster comb. Anybody, if that's the only place you could get it, would you use it in your human embryo media?
I didn't know. Yeah.
Anybody else? Thank you. Anybody in the audience, would you use it in your media if it was harvested from rooster comb? Comb, yeah, the comb on their head. That?
Trypsin was from cattle and it's also used in semen analysis.
Okay, good point. Thank you for that point. That's where I was going. Is the audience say anything? 50 yes, 60. Wow, okay, great. Man, this is really good. Because when I looked it up and I was super excited because my implantation rates, I'm doing some blast work. I'm thinking like, man, what do I got to do? I'm gonna get some glycosaminoglycan and I'm gonna make my own media. I made all my other medias in the lab, so why not make this? back in the day, we used to have to make all our cryo medias, even made our own micro tools and things. So we have kind of a 60-40 ratio here. So I called up Jacques Cohen. And I said, hey, Jacques, have you read that paper, David Gardner did? You know, he's like, I got to get me some of this glycosaminoglycan. And I said, what do you think? It's harvested from rooster comb. And he said, well, I probably wouldn't do that. So I didn't do it. But you have a good point. Hyaluronidase is harvested from bovine testicles. Chymotrypsin, which we use on our sperm, is harvested from bovine pancreas. Why can't we use hyaluronate from rooster comb? But so I didn't. I did when they finally did come out with some embryo glue. I did use that for a while. And
Tony, did he give you a reason for not doing did he give you why you shouldn't do that?
thank you, Doug. That's a good question. I think at the time there was a lot of things coming around at that time. The FDA was getting involved with more of the approval of the medias and approval of the devices. And there's a lot of discussion on that. And as a matter of fact, I mean, I didn't mention this before. Prior to these sequential medias, I actually had the privilege of working with Klaus Wiemer, AKA Dr. Co-Culture. And so co-culture is when we used to use bovine oviductal cells. He used to go to Canada every summer, harvest bovine oviducts and pull out the oviductal tissue and freeze that tissue. He'd monolayer the tissue out, the cells out. So what we were doing is we were growing cells on the equivalent of fallopian tube cells in the lab. And so we did a lot of co-culture. In fact, pre-sequential medias, we did a lot of blastocyst work, actually. We were one of the early people, even before this paper came out. This is back, I started with Klaus in 1994, actually. And we did co-culture all the way up till probably 1999. And then we started using some sequential medias. And I think it was because of that we were getting away from using animal-derived products in human work. Even the bovine serum, the BSA, we stopped using BSA, bovine seroalbumin. We did continue to do some co-culture. Actually, there's a paper out there that the Cornell folks actually did quite a bit of work. Lucinda Veek, she had a couple papers out using endometrial cell co-culture. And I did some of that when I worked at Reach in North Carolina. We would actually go in, the doc would hand me a piece they would go in and do an endometrial biopsy. They would hand me that biopsy. I'd take it down to the lab and I would treat those cells the way Klaus would, the oviductal cells. I'd plate them out, let them grow for about 6 to 10 days. And I would trypsinize those and freeze those cells in a tube. So when a patient came through, I would just thaw those cells out, put out a monolayer in the embryo. They would grow their embryos on their own endometrial tissue. So, we were just trying to find ways to make better embryos, right? So this was even after sequential medias were around. So we were probably doing this along with using some of our sequential medias. I think I did use blastocyst media on that oviductal and then endometrial tissue.
Did that improve the fertilization or the implantation rates and stuff?
But that's we've been he quotes in here 50% implantation rates, but there's still people out there today that don't have 50% implantation rates. And 50% implantation is basically saying out of every 100 embryos you transfer, half of them are implanting. And that's pretty high. I would say, my pay less than 35, I'm at the 50%. But when you get up to patients that are greater than 35, the implantation rates do go down. And so that's where could you implement oviductal cells? Does it help give you better embryos? Can embryos repair themselves? For me, embryology has been one of those things. The reason why I love it so much is that just when you think you figured it out, there's something else to figure out. And you're never going to figure it all out. And so that's kind of why we do this journal club. That's why we have the academy, and we get people to interact here. So I'm going to shut up for a little bit, and hopefully see if anybody has any comments in the audience that I can add to maybe the conversation.
I have a question.
Yeah.
Why is glucose not good for early stage embryo?
Well, the glycolytic activity is low. That's one of the things that he was saying is that, the glycolytic activity is low, so it's not really metabolizing that.
How do the amino acids help fix that?
Well, the non-essential amino acids possibly. Even if you I read this and I think that's, as a not an embryo, but as a human, I still want to take in protein. I want to take in my amino acids so I can build protein and build muscle, build brain cells, nerve cells. But you can actually do too much protein, and that will then turn into carbohydrates. So maybe too much of a good thing isn't good. So, and I've said this in other journal clubs. This was really great work that David Gardner did, but I think that, if somebody were to, the technology, we mentioned this earlier, talking about chymotrypsins and things, or the DNA fragmentation, which has been another topic here. I think you could take some of these early papers and redo them, and I think you could really drill down even closer to what is in the fallopian tube, what is in the medias, what is it. He doesn't mention this in this paper, but I'll see if I can find that old paper. But he has where not only glucose and the EDTA and the amino acid, but he talks about what amino acids, specifically what amino acids are being consumed and produced by the embryo. Like sometimes you put in this much amino acid and there's more of that amino acid being released into the media. There's a lot of work around now on metabolomics. What's in the media? I don't know that they're looking at specific amino acids, but so yeah, I'm gonna open it up to the audience. Do they have any questions, comments?
Hi.
Um,
I just wanted to go back to like the embryo glue thing because I
Yeah.
did used to work in a lab. They would like either use embryo glue just in very specific patients, like when the doctor requested embryo glue, but they do used to supplement their transfer media with 30% of HSA, which I thought was very interesting. The justification for that was like my lab director said that it made the media stickier. So the embryo would like stick to the endometrium basically. And now that I'm working in this other lab and we don't use that and we use the culture media to do the transfer, I am actually seeing a higher implantation rate. And it makes sense to me because it's like you're not changing too much the ambience of the embryo. So like you give it like a little bit more of like a cushion so it can make like an adjust and stick to the endometrium. But I don't know what you think about that, like supplementing the media for like 30% with HSA.
So you're seeing higher implantation rate without the embryo glue and the high HSA?
Yeah.
Okay. Well, it's a great question. There was a time when we used to do our transfers, we used to use 50% maternal serum. So I told you we used to obtain the serum, and I had a refrigerator full. If I had 10 patients this week, I would probably have, in 10 patients last week, I'd have 20 tubes of serum in there. And so when we were setting up for your transfer the next day, we would take that serum, and hopefully we had enough left over to do your transfer. We would mix it at 50%. And so my personal opinion is I don't have any data to support it other than the fact I don't do that anymore and my pregnancy rates are very good. Um, and, but, I think that it probably doesn't actually harm it that much, but the idea is that the serum is not really meant to, it's not really meant to be growing in a serum environment. I think if you're going to use embryo glue, you should use it the way they recommend. I used embryo glue and I didn't use it in their recommendation. I would just put the embryo in and leave it there for, I think he even says here in the paper, for 5 minutes. And that's why he says if you leave it in there for 5 minutes is the equivalent of growing the embryo the whole time in the hyaluronate media. And it can be very expensive. So why pay for the hyaluronate to grow as your protein supplement? Just put it in for the last 5 minutes right before you transfer it. And that's what I would do. I would move the embryo into that tube, into that drop with the hyaluronate, and I transferred it. I work with some labs that I've done some offsite consulting with, directing in, and they swear by it. They use it all the time, but I've never heard anybody using the high protein supplementation. That was something we did back in the 90s and pretty much got away from it when these sequential medias came out. So yeah, thanks for that comment. Appreciate that.
Yeah, thank you for
Anybody?
answering.
When it comes to implantation and pregnancy rates, it doesn't matter what media you use, that transfer quality is probably the most critical piece of that, in my personal opinion. Because you got a great embryo, you got good media that's been holding it all the way through up to day 5. So when it comes to the transfer, it's really important that the doctor gets it in the right place at the right injection speed without causing trauma in the uterus. And if that happens, you'll have high implantation rates. Actually, in my newsletter, my next newsletter is going to address that off of our LinkedIn page. So follow us on that. You'll get some other nuggets and tips. This past week, I talked about pH, osmolarity, temperature. Those are the 3 important things we have in the lab. All right, so
You was talking about implantation rates, but like if a little bit of oil is accidentally still in the catheter and put in the uterus, should the patient gonna have cramps, right?
yeah, mhm.
It's not gonna.
Yeah, the cramps. Yeah, the cramps. When they have cramps, there's what is it? It's not oxytocin. What's the progesterone supports pitocin. But you have the cramps, and what that does is it causes it's kind of like having the corticoids are one going to go up, and two, the the cramping is kind of what you'd have during a period when you're trying not to shed, when you're shedding the lining. And so I remember my first transfers in the 90s. I never saw a patient get a transfer without crying. The catheter was about as rigid as this pen. Imagine poking that through a cervix. Everybody cried. The fact that we had 10% implantation was pretty amazing. But then the Wallace catheter came out, the soft noodly catheter that you guys are practicing with, and that the implantation rates went through the roof with that. Just not causing trauma in the transfer. I say through the roof. We went from 10% to say 30%.
But it's good.
Yeah, that's pretty good. Today, and so that was what was really great, is that he was saying that 50% implantation. That was a big, bold statement back then. So kind of stepping forward, we talked a little bit about this. We're now using, I think everybody in the audience even said that they're using the single step media. Sounds like everybody's buying it pre-supplemented. I like doing that because nobody is making any errors in the lab and they're mixing. When you ask someone to make a 10% solution, it's really funny what you'll get. You can have one mil of serum to 9 mils of media to 0.1 mils of serum to 9 mils of media. Like I've had all kinds of different answers in that. So my biggest reason in doing that is because nobody makes a mistake. And I'll just going back to where, when they started making pre-supplemented media, Pat Quinn actually called me up and said, "Hey, Tony, you want to try this media out for me? Just, keep your data, so, track your data and see what happens." I thought it was a great marketing strategy because I was using basically my non-supplemented media with my protein that I've quality control tested, I've endotoxin tested, I've mouse tested. I got a whole year's worth because I know it's making the best embryos. And he sends me this media with no protein I don't know anything about. And I compare the two, I run sister eggs compared to it, and I had significantly more embryos from that pre-supplemented media than the media I was using my own protein for. So I don't know if you knew that ahead of that it was going to happen, but I haven't. I switched to the pre-supplemented media and never looked back. Here's another good poll question: Is everybody doing does anybody change into fresh media on day 3? Like, yes or no? Fresh media on day 3. If they're using the one-step media, do they change midway through into refresh their media? Okay. So it looks like a majority of people are not refreshing their media. So that was something, once I got to the one-step media, I was afraid to go all the way out to day 5 or day 6 in that media because, there was actually, it doesn't mention it in here, there was always some fear that there would be a buildup of ammonia from the amino acid breakdown. And so that's why we refreshed our media on day 3, even if we were using a single step media. I actually talked to a colleague of mine. I think it was Harold Rodriguez. He says, I'm just growing all the way out to day 5 and day 6, so I started doing that. I even ran some sister eggs comparing it, and it didn't see any problem. So what I'm doing now actually is I'm actually putting in that culture media on day 0 on retrieval day, and I go all the way out to day 6. Even in my dry incubator, I'm actually culturing those embryos all the way from day 0 to day 6 in the one dish that I did the retrieval in. And so I'm only using one dish for every case. I retrieve in that dish and then I culture in that dish later in the day, all the way out to day 6. So the traditional lab will typically do a retrieval day and then they move it into fresh dish on embryo day 1. And then day 1 to day 1 to, so it sounds like the majority of the audience here is doing day 1 to day 6, all the way in that, possibly even day 7. Pretty sure somebody in here is doing some day 7 work. So, something else that was that's mentioned in here is, and I know Rusty Poole here in town was actually part of the development of this media. Not Vitrolife, but Irvine Scientific had a blastocyst media, but it only had one amino acid in it. Anybody see flaws in that? Why?
Just one, especially when you're like growing cell.
Yeah, I never understood it. I did some consulting and I would go to the lab and we made beautiful blasts, had great pregnancies with it. But what they did was, and I wish Rusty was here. He could talk to us about it. But he said, like, the way I understand it, I could be wrong, is that they tested the amino acids individually with mouse embryos, like all the essential and non-essential amino acids, and said this one amino acid gave them the best blast development. And that's the one amino acid they put in the media. I forget, I think it was tyrosine. I think that was the amino acid they put in there. And so, yeah, they called it P1 media. So there was the early cleavage media, and then you had your blast media that was the P1 media. And you might even still be able to buy it. I'm not really sure. That would be with Fujifilm now is now with NextSpring. But so yeah, you think we've come a long ways. And so what do you think is in the single step media? You think there's glucose in it?
Yes.
I think there's essential and non-essential amino acids. Yeah, so basically has everything in it. I actually don't think it I think it does have some glucose. I think it does have glucose. Might have to look. So the idea is that if the concept now is that if the embryo needs it, it's there. Maybe one embryo, the genome is activated before another one. But if those essential and non-essential amino acids are needed, then it's there. When I was in college, I took this agronomy class, and I think this resonates with me with embryos because it's a lot like growing plants. I don't know why I look at an embryo like a plant, the root and the fruit, because you can take one seed and grow that into a stalk of corn with multiple seeds on it, right? I think that's just the most amazing thing ever. But if you take a plant and grow it in a insufficient soil, it's only going to grow. Like for some reason, my corn only gets this tall, and then it doesn't die. It actually tries to make corn, even though it's this tall, but should be this tall. And so it's a great analogy in the sense of if you don't have the right nutrients in your soil, the plant's only going to grow so far. And our embryos will do the same thing. And that's what they showed us in this agronomy class. They actually used a wine barrel analogy, that the wine barrel has all these oak pieces of wood in there, and they're held together by a ring. And that if one of those slats of oak only goes up halfway into the wine barrel, that barrel can only be filled half full. And so the idea is that maybe that is tyrosine, or maybe it's alanine, or maybe it's the protein, or maybe it's the lactic acid or the pyruvic acid. If one of those things aren't there when it needs to, that embryo is only going to grow to that point. It's going to stop growing. And just like a plant will, and that's the way a lot of our early medias were. They only had enough to get it to a certain point, and then they wouldn't grow. We still made blastocysts. He mentions that here. Blastocysts still could be made, but they were inferior blastocysts. I think and my early experience with blastocysts, I told you my first 10 thaws with frozen thaw blastocyst, I was 0%. And I was ready to give up on blastocysts. Thank God I didn't. And I think here at the very end, he talks about lactic acid too. I actually highlighted it here in the paper that he said blastocysts which exhibited a pattern of glycolytic utilization similar to that of embryos developed in vivo had development potential of 80%. But if those blastocysts exhibited excessive lactate production, so that's basically looking at the media, excessive lactate production, the developmental potential was only 6%. So what he's doing is he's talking about here about viability assay, like things that you could do to test. And so if you could find a way to test the lactate in your media, I think that'd be a pretty cool assay. Just test the lactic acid in your media, lactate. If you had a high level of lactate, it'd probably be an indicator that's a poor quality embryo. It talks about glucose uptake too. It's like if it's greater than 5 micrograms per hour, that it was better than if it was less than 5 micrograms per hour. So these are things that he doesn't, in this one here, he doesn't talk about the amino acids so much consumption production, but he's saying the glucose and the lactate. So those are just interesting things to consider.
What if the lactate just mean it's using up more glucose?
Probably, yeah. So what he's saying here is that if it has a high glucose uptake, it's good. And also, it'd be the inverse. It'd be low lactate would mean increased potential, right?
Yeah. The glycolytic pathway goes from glucose to lactate.
Right. Yeah, but that's the end product.
Yeah.
Right.
So it's kind of like
exactly. All right, we're coming up on the hour. Anybody have any questions, comments?
I have a comment. Anyone who's on this call today is welcome to save 25% on their next lab certificate course. So please use that code, go online and register today. Additionally, please don't forget to register for our next session on the risk factors for mismatches in the ART lab.
Great. Thanks, everybody.
Thank you very much.
See you in a couple weeks. Thank you so much. Lovely. Thanks, everybody.
Thank you.
Thank you.
Right. Good times.